We researched how the abiotic factor of temperature will potentially affect Brassica rapa distribution in the future. B. rapa belongs to the Brassicaceae mustard family. B. rapa is a winter annual. This means that it will germinate in either the fall or summer, grows to adulthood during the winter, and dies in the spring. B. rapa has a widespread distribution in the United States, as it can be found in every mainland state. However, larger populations are found in West Coast states rather than East Coast states. As the average temperature of the Earth increases, many organisms are going to have to adapt their temperature tolerances, shift habitats to cooler areas, or die out. The Earth's average temperature is expected to increase another 2 to 3 degrees Celsius within the next few decades. The temperature tolerance of the Brassica family has been studied extensively given their importance as a food source. It has been found that a cultivated version of B. rapa has a higher temperature range tolerance rather than its congenic B. napus. Research performed by Lee et al. Revealed that cultivated B. rapa, despite showing signs of stress, does survive in cold temperatures as well. Our experiment differs from prior experiments because we are focusing on wild rather than cultivated B. rapa, and potential distribution impacts rather than just survival. Based on previous research and distribution maps, we hypothesized that wild Brassica rapa has a wide optimum range of tolerance for temperature.
We tested our hypothesis by placing B. rapa plants into 3 separate temperature conditions. The thermometers consistently read 12.4 degrees Celsius for the cold condition, 22.8 degrees Celsius for the room temperature condition, and 30.2 degrees Celsius for the hot temperature condition. However, it is important to note that for the first 2 days of growing, the incubator for the hot temperature plants was set at 35 degrees Celsius. The cold and hot temperatures were chosen based on average October temperatures of Maine and Florida, 2 states where B. rapa grows, demonstrating the plant's wide distribution range. Given the time limit of the project, we used Wisconsin fast plant seeds to give us quick growing time. We planted 32 seeds per condition and allowed them to grow for a total of 16 days in the hopes of them reaching flowering stage. We used a watering apparatus and automatically watered the plants using felt cloth and wicks that drew water up by adhesive and cohesive force. As long as the container was filled high enough with water, the plants stayed watered. To control for light variable, we used 2 incubators and a fluorescent lamp with timers set to 12-hour daylight and 12-hour darkness to stimulate fall daylight length. For the room temperature condition, we covered the plants with a box because we had no third incubator. Throughout the experiment, we measured height in millimeters daily. We kept track of germination and survival as well to determine germination and survival percentage at the end of the experiment. We also counted true leaf number of every individual plant starting at day 12. Finally, at the end of the 16-day growing period, we uprooted our plants and set them in a drying oven for 24 hours at 90 degrees Celsius. This allowed us to collect dry mass data, which we measured in grains.
To analyze the height data, we took the average for 2 days for each living plant and then averaged those numbers together to create the overall average height for every 2 days of growth. When comparing the height change over time, we found that the plants in the 30-degree condition shot up fast before reaching a plateau. The plants at room temperature eventually surpassed the hot plants. The cold temperature plants did not germinate at all until day 5 or 6, and then they slowly grew at a relatively stable rate. Standard error was very low for all of this data. True leaves did not grow and were not counted until day 12. Only data from live plants was analyzed and the room temperature plants appeared superior. By the end of the experiment, the cold plants were almost equal with the hot plants. For dry mass, every plant that grew was dried and measured. The room temperature plants showed higher overall mass and the cold plants were heavier than the hot plants. No statistical analysis could be performed on the germination percentage and the survival percentage data, while the hot population experienced the most death.
We found that B. rapa plants germinated in a wide range of temperatures varying from 12.4 degrees C to 35 degrees C. The plants in the more extreme temperatures were not as long-lived nor as healthy as the plants in the room temperature range. We found that B. rapa appear to grow best around room temperature, 22.8 degrees C. Without performing t-tests, we cannot say that there is a significant difference between the groups. Our data appeared to support our hypothesis in that the plants grew well from 12.4 degrees C to 22.8 degrees C. However, above that temperature range, they quickly experienced stress and did not grow well. Lee et al. Shows that the cultivated B. rapa plants experienced delayed germination when in cold temperatures, which we saw. However, they also say those plants exhibit poor growth and poor head, which we did not see in our plants. Mgadi's research shows that B. rapa experiences much stress at 28 degrees C and above. Our data gives more insight into the impact of high temperatures on B. rapa growth. The damaging effects of consistently hot temperatures could cause the distribution of B. rapa to move northward and the range would be more limited.